As many of you know, antioxidants are touted as health benefits, but oxidation is the opposite of antioxidants, and the greater the oxidation reduction potential in a particular species, the greater stress that plant is under. So by using the deluxe analyte package, you can access further and deeper insights into how your plants are performing, or actually act as an additional layer of confirmation that the data that you're getting from a mineral perspective is also accurate and true. And when we've coupled our deluxe analyte package with our basic mineral package, we consistently see this theme. And this is something we've spoken about in previous episodes, is that nutrient stress precedes plant stress vulnerability. The protein denaturation, cellular dehydration, and cell wall compromise are huge drivers of plant susceptibility to a wide range of insects, disease, weather stress, and unwanted crop outcomes. These are not isolated to an entire plant. They can be isolated to a specific plant organ, such as a leaf, or a bud, or a flower, or a fruit. The amount of carbon that the plant can access in relation to the growing habitat is directly proportional to the plant's ability to turn on its own immune system. All of these stress points can be limited through proper nutrition, and they are also reinforced through improper nutrition. So I want you to think about that for a minute. So in other words, our bad habits or our unseen mistakes can reinforce protein denaturation, cellular dehydration, and cell wall compromise. And so when a plant has too much food, not only does it take money out of our budget, but it starts to clean all the overfed cells. These special proteins then are upregulated within the plant's own vascular system, like I mentioned a minute ago, about upregulation of massive amounts of protein. These fortify the plant's long-term health in order for the plant to survive. But during that process, the plant also sequesters all of the dead cells and dissolves them as best it can so that it can resume its normalized growing state. So what you have is a plant that takes all of its toxins from the new leaf, oftentimes, puts them into the old leaf, sequesters them there until the plant can drop that old leaf on the ground and feed it back to nature. This is something we all see year over year, whether it's from additional chloride stress or sodium stress or metal stress or some other form of fertilization. There's oftentimes a leaf drop due to leaf senescence. Similarly, we've seen where a growing tip of a plant can sort of peter out and start to curl up and pinch, and those are often indicative of a different nutrient excess that a plant can't get rid of. And so the plant will stop growing vertically and start to grow horizontally instead. This helps the plant to prevent sick cells from reinfecting itself, and it helps the plant to shed unhealthy cells. Why is this important? Because common natural excesses such as aluminum, sodium, chloride, or manganese in certain situations, or boron in high boron areas, or even calcium in high calcium areas, they're almost always correlated with physiological symptoms on our leaf analysis. We also see induced excesses from fertilization standpoints due to overapplication for young growth plants, so where we try to load up a large amount of fertilizer at a early growth stage where the plants are really young and small to save us on labor or something like that. Oftentimes we see induced excesses causing similar problems. Some of the items listed here, such as poor fruit set or nutrient lockout or poor sugar development, all of these things have consequences economically, seen or unseen. But as many people have realized, that a lot of these problems start in mineral quantitative data. And so if we find high aluminum availability on water-soluble extracted soil analysis, even at neutral or high pH, or if we find high sodium in leaf analysis, we find huge amounts of soil depletion that are happening. When there's high magnesium to calcium ratio in a leaf analysis, that soil has depleted calcium and most likely has a depleted calcium to magnesium ratio, or a overall base saturation calcium that's less than desired. When we find high aluminum to phosphorus ratio in leaf analysis, we see that as an indicator of soil mineral depletion. And last but not least, we find that when calcium to magnesium ratios are inadequate, as Dr. William Albrecht confirmed many years ago, that soil compaction and lack of soil aeration, flocculation, soil aggregate stability, etc., is an outcome there. When we have abiotic visible stress in quantitative data, we often have low levels of key nutrients, such as phosphorus, calcium, silica, molybdenum, or boron. We see imbalances within these cause visible physiological disorders, such as seed viability or leaf tip necrosis. Oftentimes, these physiological disorders have vulnerabilities that get exploited by insects or disease. When we see disease pressure and its correlation to mineral quantitative data, again, calcium to magnesium ratio has potential. Aluminum to silicon ratio has potential to be a problem. And oftentimes these correspond to regional nutritional imbalances. When we see insect pressure in mineral quantitative data and the corresponding data sets, we often see a imbalanced aluminum to phosphate, aluminum to silicon ratios. We see imbalanced calcium to magnesium ratios. And we also see, again, regional nutritional imbalances associated. When we find growers that have crop failure in mineral quantitative data, we find massive excesses, typically during reproductive phases, of aluminum, sodium chloride, or all three. We often see corresponding calcium, silicon, and total nitrogen deficiencies during key growth stages. We have a relationship between all of these to carbon deficiencies. And so I want you to circle back to the Mulder's chart that we've looked at in previous episodes, the one that looks like the dream catcher. But if you take carbon and just smack it right over the top of that one, carbon relates to every single mineral's availability. And so we've been operating under a paradigm that has us going in circles trying to balance nutrients when in reality there's a key nutrient that's just missing generally, a key macronutrient that's missing. Carbon is king. Thanks again very much, and look forward to seeing you all next week. Take care.